Okay, gonna get going here. So, can you hear in the back? Yeah. All right. So, what we're going to do today is talk about what I call acquisitions and mergers. And we've already discussed sort of a little bit of background to this, but we're going to go in a lot more detail into a couple of examples of it. And then in the next lecture later today, we're going to talk about extremophiles. Just as an aside, I have a project run out of my lab in collaboration with a lot of people that is sending some microbes to the space station and then going to be sampling the microbes that live on the space station. The launch is later today. This is like the 8th try at a launch. It's been delayed a million times, but I have a bunch of people from my lab as well as other students from UC Davis and collaborators who are in Florida right now waiting for the rocket to launch to send samples up to the International Space Station. So if you want to follow, it will be broadcast live on NASA TV later today, and you can follow on Twitter at Space Microbes hashtag. We wanted to have it be, microbes in space, but that was a little too long for a hashtag. All right, so what we're going to do today is go into a lot more detail than we have already about the history of mergers and acquisitions in two different aspects of this. So in eukaryotes, there are a large number of examples of where a eukaryotic cell has brought another organism inside that cell. That's generally known as endosymbiosis, and that's behind the history of the mitochondria and chloroplast, and it gets complicated in terms of the history of the chloroplast a little bit. It's also, as we will see later in the course, behind the biology of many plant and animal functions that they've engaged in symbioses with various microbes. And then we're going to talk about another form of acquisition where organisms take, rather than bring an organism inside of them, they can bring the DNA from another organism inside of them and merge it into their genome. And that's known as lateral gene transfer. And these two processes are both examples of cases where drawing a simple evolutionary tree with bifurcations, that is, two branches coming out of a node, is not sufficient to represent the complicated web of life, the evolutionary history of life that actually has occurred. And I'll try and highlight these as we go through them, but there are sort of four key concepts related to the lecture today. One of them is that we've already sort of talked about this. Different parts of a cell or genome can have different histories. Another is something we've also referred to, at least in terms of the organelles, but the history of eukaryotic evolution contains many, many examples of these endosymbioses, bringing another cell inside of the eukaryotic cell. And in contrast, the history of bacteria and archaea, at least as hosts, that is, as recipients of foreign material, there aren't many examples of bacteria and archaea bringing other cells inside of them. There are a couple, but it's very, very rare. But there are thousands upon thousands of examples of bacteria and archaea bringing DNA from the outside, from a different species, from a very distant relative in some cases, into their cell. And then the last point, which is we'll sort of cover throughout the lecture, is that theories about evolutionary history, which we will discuss today, they can be tested explicitly with analysis of data. So we're not, in this class, just going to tell you this is what is known about evolutionary history. What I've tried to do and what we will continue to do is say this is how this particular aspect of evolutionary history can be tested. And you can distinguish this model theory from another model about evolutionary history. So we're going to go through one case first. which we've already talked about briefly, and that involves the evolution of the mitochondria. So the mitochondria is this membrane-bound organelle inside eukaryotic cells involved in particular, and many of them, in ATP generation via various metabolic pathways. And we're going to present I'm going to present one theory of mitochondrial evolution. I've already talked about this theory before, but then we're going to talk about what the evidence is for this theory and how you might test it. So the theory goes back to something I've already talked about, which is, imagine there was an ancestor of the eukaryotic cell in some branch in evolution, probably after the common ancestor of the Archaea and the eukaryotes split. This cell had its own genome, and this cell engaged in a symbiosis with a free-living bacterium in the group, the Proteobacteria. This is one of the lineages of bacteria that had already evolved and diversified. And there was some type of interaction, tight association, could have been mutually beneficial, there could have been a negative relationship here, it's not entirely clear, but they were closely partnered and starting to interact with each other. And at some point, this host cell used a flexible cell membrane and the process of phagocytosis, where they use this sort of enveloping of particles on the outside to feed, to wrap its membrane around this bacterium until it was completely enclosed. And then that becomes now inside the cell. So here's the bacterium inside the cell, surrounded by a layer of the cell membrane. Once that bacterium is inside the cell, it's sort of generally referred to as endosymbiosis. Symbiosis means living together. Endo means inside. So this is an example of an endosymbiosis, when one organism, that is, in this case, the host, brings another organism, that is, the symbiont, inside of its cell. And after this happened, in many cases, eukaryotes do this all the time. Amoeba do it, our white blood cells do it. Lots of the eukaryotic diversity does this all the time to eat. They eat other cells. But for some reason, this cell survived inside this other cell and eventually became a mutually beneficial partnership that led to this cell throwing away some of its components. Oh, sorry, before I go on, this is when an organism is brought inside another organism. If that symbiont has never experienced a symbiosis like this before, it's called a primary symbiosis. And this is going to be important because we'll see other kinds of symbioses. So this is the primary symbiosis in the history of this proteobacterium. So it's been brought into the cell, it starts to evolve this sort of mutually beneficial interaction. At some point, the DNA of the host cell gets compartmentalized into a nucleus. It's, as I mentioned before, it's unclear if that happened before this symbiosis occurred or after. We can't really resolve the order of these events very well. At some point, that bacterium loses some of the features that are typically found in free-living proteobacteria. So it throws away its cell wall, for example. It throws away some of its cellular functions, some of its membranes, but it still retains a membrane around it. That is a membrane that resembles the membranes found in bacteria. And it now has the host membrane around it, and it still retains at least a small amount of its own original genome. So now you have two genomes inside this cell that have different evolutionary histories. And that cell, this cell here, the model, I'm still going through this hypothesis, this model about mitochondrial evolution. The model is that that cell was the common ancestor, a cell like that was the common ancestor of all the eukaryotic lineages that we observe today. Those 7 major lineages, I'm only showing 5 here, but those 7 major lineages that I went through in the last class, this cell with a mitochondrion that came from a proteobacterium and a nucleus was the common ancestor of all of these lineages. That's the model. And the model is based on a variety of pieces of evidence, but I'm going to take you through just a little hint at how you might test this model and distinguish it from other possible models to explain the observation of these bacterial-like organelles found throughout eukaryotes. So one prediction of this model is that if you look at genes in the nuclear genome and build an evolutionary tree of those genes, they should look something like this, with eukaryotes all grouping together, eukaryotes potentially being the sister group to Archaea, and then bacteria being the outgroup relative to them. That's our model of the 3 domains of life. But if we look at the genes in the mitochondrial genome, this is a piece of DNA inside the mitochondria, but that mitochondrion is inside a eukaryote. So it's, in essence, studying a different part of the genome of a eukaryote. This part is contained within the mitochondrial organelle. If mitochondria came from bacteria, the evolutionary tree of mitochondrial genes should look like this, where they should group within the bacterial branch of the tree to the exclusion of other bacteria and should not be a sister group to Archaea for those particular genes. Does this make sense? Are there questions about this? Okay, so that's our model. So let's consider an alternative model. What if all of the mitochondria that we observe in modern eukaryotes, what if they had multiple separate origins? Say the mitochondria in plants separately evolved from bacteria from the mitochondria in animals. What should the evolutionary tree of mitochondrial genes look like then? It should look something like this. You should have the mitochondrial genes that are in eukaryotes showing up in many different places in the prokaryotic tree. Maybe some mitochondria could have even come from Archaea. Those ones would group like this in the tree. Maybe some mitochondria came from one lineage of bacteria and some mitochondria came from another lineage of bacteria. The tree would look something like this. This is just a model here, but we now have 2 alternative hypotheses that give very different predictions as to what phylogenetic trees of mitochondrial genes should look like. And this is basically how we test hypotheses about the evolution of these features. We propose a model, we propose alternative plausible models, and then we test them by looking at the actual data. So how do we test these models? Well, we talked about you could look at mitochondria and try and build an evolutionary tree of the different mitochondria based upon their phenotypic appearance, their morphology. That doesn't work well because morphology for bacteria and Archaea is not a very informative trait. There's lots of homoplasy for this trait. Lots of distantly related bacteria have similar phenotypes. So we can't use just their appearance to tell us where they came from. Fortunately, mitochondria still retain DNA inside that organelle. And we can take that DNA out and study it just like Carl Woese took ribosomal RNA, or eventually the genes for ribosomal RNA, out of cells and built evolutionary trees. So this is going back to what I showed you with the model for how Woese studied evolution of bacteria, Archaea, and eukaryotes. We can do the same thing with mitochondria. Now we can add their data to the data collected for other organisms. You can actually pull out ribosomes from these. You can look at the ribosomal RNA. Most people now, we'll talk about this later in the class, look at the DNA that codes for the ribosomal RNA now instead, but the principle is the same. Sequence it, build an alignment, a data matrix of those sequences, and build an evolutionary tree. And this is what the tree looks like. All mitochondria are monophyletic. They trace their ancestry to a single common ancestor mitochondria that evolves from within one branch of bacteria. So this supports the original model I proposed, one event prior to the diversification of all eukaryotes, and all eukaryotes inherited the mitochondrion from that event. This is actually real. What I showed here is sort of a model, hypothetical data. This is actually real data. This is what you end up getting when you build one of these trees. Here's a group of bacteria with different lineages of bacteria, including different kinds of proteobacteria, chlamydia, spirochetes, and then alpha proteobacteria, and within the middle of the alpha proteobacteria are all of the genes that come from mitochondrial genomes. So this is most consistent with that model that I first proposed. There are alternatives. You have to continually test alternatives, but none have been proposed that have yet held up to scrutiny over the model that I proposed there prior to the origin of eukaryotes. So basically what this tells us is a couple of things. It tells us when this mitochondrial symbiosis event happened. It also tells us that the mitochondrion has a different evolutionary history than the nucleus. The coloring here is backwards. Sorry about that. That should be red. So the mitochondrion tracks with this tree. The nuclear genes track mostly with this tree. I mentioned, and I'll come back to either at the end of today or the beginning of tomorrow, some genes move from the mitochondrial gene into the nucleus, the mitochondrial genome into the nucleus. Those still retain the evolutionary history of mitochondrial genes, and you can find them now in the nuclear genome of eukaryotes. So eukaryotes have at least two distinct evolutionary histories represented within their individual cells. There are many other implications of this model of mitochondrial evolution happening at the beginning of eukaryotic evolution. I'm not going to talk about them in detail now, but you should think about them maybe for a test. So what does this model imply about the evolution of eukaryotes that do not have mitochondria? That's a question to think about that might show up on some exam in the near future or something related to it. And another question is, what if you took out mitochondrial genes from, say, humans, chimps, rats, and birds, and you built an evolutionary tree of those mitochondrial genes, and you compared them to an evolutionary tree of nuclear genes from the same species, ones that had not undergone any type of lateral transfer. What should the evolutionary tree look like once you're looking within eukaryotes? That is, for mitochondria, do humans and chimps, should they be most closely related to each other to the exclusion of, say, mouse, and versus nuclear genes? How do mitochondria and nuclear genes co-evolve with each other after this symbiosis happened? Another thing to just think about. So we're going to switch over from mitochondrial evolution to now talking about chloroplast evolution and apply the same general principles that I just did for mitochondria. It's going to get a little more complicated, but the same principles apply. So we're going to start with a model of chloroplast evolution. The model is very similar to the model for mitochondrial evolution, but the model proposes that the starting cell for this symbiosis already had a mitochondrion and already had a nucleus, so it's at least a little bit later in eukaryotic evolution than the mitochondrial symbiosis. This eukaryotic cell interacted with a free-living cyanobacterium. Cyanobacteria are one of the phyla of bacteria that we talked about previously. They are photoautotrophic. All representatives that have been studied in detail are photoautotrophic. That cyanobacteria was engulfed by this phagocytosis process, brought inside the cell. This is now another primary symbiosis event. The primary term refers to the history of the symbiont. So in this case, the cyanobacteria had not been involved in a prior endosymbiosis. So now we have a new primary symbiosis for this group that is bringing in the cyanobacteria, which eventually became the chloroplast. Starts to throw away components of the cell, just like the mitochondrion did. As I mentioned, in some chloroplasts, we still see peptidoglycan, so not all descendants of this evolutionary event threw away everything from the cell. Some of them threw away the outer membrane. Some of them threw away the cell wall. Some retained it. But eventually, these became the chloroplast. So now we can ask if this model is correct, again, just like with the mitochondrial model, if this model I proposed is correct, what should phylogenetic trees of chloroplast genes look like? So what should phylogeny of chloroplasts look like? Again, like with the mitochondrion, DNA is a much better tool for studying the evolution of chloroplasts than phenotype is, because phenotype is misleading. So if it was a single origin, what should the tree look like? This should be what the nuclear genome of this cell looks like. And just like with the mitochondrion, if the chloroplasts all derive from a single ancestor, they should all track their origin to one point. They should be monophyletic. They may group in a different part of the tree than the mitochondrion, but they should be derived from a single event of a symbiosis with some type of prokaryote, at least. What if the chloroplast had multiple origins? Again, it should look like this much more complicated model with chloroplasts grouping in different parts of the tree of bacteria and archaea. This is what I already mentioned. So to test the models, again, phenotype is not going to be particularly informative. We turn to DNA. Chloroplasts still retain some of their genome left over from their free-living days. And if we build an evolutionary tree of chloroplasts, what we see is that they are monophyletic. They all share an ancestry with each other, and they evolve from within the Cyanobacteria. So it's a single evolutionary event of a symbiosis with a Cyanobacteria that led to all modern chloroplasts. We can zoom out just to show you what this looks like. If we zoom out, there are, again, hundreds of lineages of bacteria. So if we zoom out, this is from within this one lineage here, the Cyanobacteria. Sometime in evolutionary history, one of the branches of Cyanobacteria eventually became the chloroplast. So now if we look at the history of cells again, here we have cells with a mitochondria, chloroplast, and a nucleus. Again, the mitochondria should be red here. Now we have three separate origins for the DNA inside this particular type of eukaryotic cell. I didn't draw it, but just like with mitochondrial evolution, chloroplast DNA also moves to the nucleus. So the nuclear genome has genes that have the normal nuclear ancestry, genes that have mitochondrial ancestry, genes that have chloroplast ancestry, and then inside these cells there are mitochondria with genomes and chloroplasts with genomes. Getting to be pretty complicated evolutionary history here for those organisms that have chloroplasts inside of them. So, fortunately or unfortunately, depending on how you look at it, trying to apply the same follow-up model that we did with mitochondria to chloroplasts does not work well. That is, we can propose that this symbiosis happened at the beginning of eukaryotic evolution also, maybe after the mitochondrial evolution. You could even propose it happened before, and that all of the major lineages of eukaryotes inherited a chloroplast. But this model is inconsistent with much of the data that we observe, or it's hard to fit this model with many parts of the data we observe. So, for example, if you go look at all of the eukaryotic lineages that are out there, we actually see, zooming in on here, this is an old evolutionary tree from one of the older versions of the book, but it still basically works. In this case, they had 5 major lineages of eukaryotes instead of 7. Don't worry about those exact details. The point here is to show you that if we plot at the tips examples of eukaryotes with chloroplasts, there's a much more sporadic distribution pattern than we see for mitochondria. Some lineages have them all, some lineages all the members have them, some lineages none of the members have them, some lineages just a couple of members have them. So if to fit this model with chloroplasts evolving at the beginning of bacteria, we have to propose many chloroplast loss events to get to organisms that don't have the chloroplast. Probably hundreds of these loss events, and it's just not the simplest model to propose that. Still possible, still can test that model, but it's a little more complicated than the mitochondrial evolution model. An alternative model has come up, proposed by many scientists, that is much more consistent with the data, and I'm going to show you this. And it's based in part on observations of organisms like this very strange species called a cryptomonad. You don't have to worry about what it is. But if you look inside a cryptomonad cell, you actually see a regular nucleus, a regular mitochondrion, and then this convoluted structure with many, many membranes surrounding another nucleus and a chloroplast. And this is very strange, so it has two nuclei. One of the nuclei is contained within a compartment with the chloroplast, surrounded by like 5 different membranes, I believe. And it turns out many other eukaryotes with chloroplasts look something like this. And this has led to the proposal of an alternative model to explain the evolutionary history of chloroplasts. And the model goes as follows. The primary symbiosis with chloroplasts happened only in the common ancestor, the lineage leading up to the common ancestor of the plant group, the Plantae lineage. So it did not happen at the origin of eukaryotes. These different Plantae lineages all got chloroplasts. They then evolved and adapted particular features for their photosynthesis system and even for the chloroplasts that they have. So glaucophytes had one particular model, red algae had another, chlorophytes had another, land plants and charophytes had another. There's diversification within the Plantae lineage as to what the chloroplasts are doing and what these cells look like. And then, bizarre, but this is the model is that other eukaryotic lineages then brought one of these Plantae eukaryotes inside their cell to generate a new symbiosis. This is called secondary symbiosis because the cell that is being brought inside already underwent its own symbiotic events with primary symbiosis. So I'm going to take you in a little bit more detail through this model. So here's the model like I was drawing before. Imagine a normal sort of eukaryote from any of these other lineages that don't have chloroplasts. Should have a nucleus and a mitochondrion. It engaged in a symbiosis with a single-celled member of the Plantae lineage that had a nucleus, a mitochondrion, and a chloroplast. It was engulfed, brought inside the cell by endosymbiosis. Again, that's a secondary endosymbiosis because the symbiont already had undergone primary symbiosis. Over evolutionary time, perhaps they throw away a mitochondrion. That may be why the example I gave only has a nucleus and a chloroplast inside this other compartment of the cell. Sometimes maybe they throw away the other nucleus, but they keep the chloroplast. And that chloroplast will actually, in many cases, retain all of these extra membranes that are resulting from both the primary and the secondary symbiosis. So if this model were correct, let's test it with some of these other lineages of eukaryotes that are not in the Plantae group, but that have chloroplasts. So here's one of them, the Euglenids. This is part of the reason why I wanted to introduce all those different lineages of eukaryotes. So here's the Euglenids, one of the lineages within excavates. They have photosynthesis via a chloroplast. Here they are over here. The nuclear genome of Euglenids tell us that they are related to excavates. They are not in the Plantae group. If we build evolutionary trees of the chloroplast genes from the Euglenids, their chloroplast group within this one branch of the chloroplast tree, which is the chlorophytes. All these other members of this group are the chlorophytes, one branch of the Plantae group. So here's the model to explain that. You had a lonely excavate cell looking for a friend. It brought inside of it a chlorophyte that already had evolved some of the features of chlorophyte cells and chlorophyte chloroplasts. And that became its symbiont. So now you have a cell that has a nucleus, a mitochondrion, and then a second nucleus, mitochondrion, and a chloroplast. And this is what gave rise to the common ancestor of Euglenids. So it had a normal eukaryotic backbone, and it brought inside of it another eukaryote that had a chloroplast. And every piece of data that you look at in the Euglena genome is consistent with this model. This is the best model we have to explain the evolution of Euglenids, this strange but true secondary symbiosis. Let's consider diatoms. So again, diatoms, they're in the stramenopiles. Many of them are, or most of them are photoautotrophic. They have chloroplasts inside their cells. Again, if you look inside diatoms, they have a somewhat strange membrane structure around their chloroplasts that is not consistent with there being a single primary symbiotic event. It's much more consistent with there being a secondary symbiosis. Here are diatoms over here. If you build an evolutionary tree of the chloroplasts inside diatoms, their chloroplast group inside the red algae portion of the chloroplast tree. So the model is that there was a lonely stramenopile in this case. It found a buddy in some red algae, single-celled red algae, that it brought inside of its cell to have a photosynthetic chloroplast, and eventually started to get rid of some of the other features of this cell. Again, another secondary symbiosis to explain photosynthesis in diatoms versus euglenids. Turns out that understanding the biology of diatoms, it's very helpful to know that they have their photosynthetic system came from a red algae rather than from, say, a chlorophyte. They have different functions, and knowing this history is important for understanding the biology of each of these different groups. So there are many other secondary symbioses that have been shown by doing this general type of analysis. Dinoflagellates, amoebozoans, apicomplexans, many others. I'm not going to go through them all here. The apicomplexan one is very interesting. We probably will talk about this a little bit in the lecture on Wednesday because they have these relic chloroplasts that can be targets for drugs that can help treat malarial infections. So again, dinoflagellates have these things. Won't let me advance my slide. Apicomplexans have chloroplasts, amoebozoans, some of them have chloroplasts. Some cercozoans have chloroplasts. Those are all apparently the result of these secondary symbioses. Now, it gets even more confusing or cooler, depending on how you think about it. If you look at some lineages of eukaryotes, we can't fit their biology into primary or secondary symbiotic histories. So for example, there's one dinoflagellate that has endosymbionts, these plastids with just gobs and gobs and gobs of membrane. You can barely count them around it, this cryptoparadigm. And if you look at the genes inside these organisms, you can't fit it very well into an evolutionary history of secondary symbioses. The same is true for many, if not most, brown algae, like kelp. And the best explanation for some of these lineages is now a tertiary symbiosis. So we had a normal eukaryote with a nucleus and a mitochondrion, and it brought inside of it an organism that had now had a secondary symbiosis, like a euglenoid lineage. So this euglena could have had its original nucleus and mitochondrion, its new nucleus and mitochondrion, and its chloroplast. The host has a nucleus and mitochondrion, gets brought in by endosymbiosis, and in theory, there could be 7 distinct genomes inside this cell. And with DNA moving between the different genomes, even when one of the genomes disappears, like when the nucleus of one of these compartments disappears, some of the DNA from that nucleus might have already moved to the genomes of some of these other compartments. So now we have 7 distinct evolutionary histories contained within one cell. There's a really cool example that appears to be a tertiary endosymbiosis involving animals. Sea slugs have a photosynthetic system, and they appear to be bringing inside of them organisms that have already undergone secondary symbioses. So basically, this sort of evolutionary studies of the mitochondria and chloroplast, the way we figured out what's going on is to start with a simple model, to test the data against that simple model. The simple model works really well for mitochondrial evolution. It works well for chloroplast evolution if you just focus on the Plantae group, but it doesn't work well for chloroplast evolution if you look at other lineages of eukaryotes, which is why the secondary and tertiary symbiotic events have been proposed. And to tease all of this apart, you have to have a model that you then compare your data to that helps you say, my data is consistent with this secondary symbiosis or tertiary symbiosis model. And that's basically, for the rest of the course, we will talk about examples where we think we know the evolutionary history of some group. In many cases, models like this were tested versus other models before you get to the point where we say this is what is thought to have occurred. Now, it turns out we'll talk about these later in the course. There are many, many, many other endosymbioses involving eukaryotic cells that are not chloroplasts or mitochondria as the symbionts. There are just endosymbioses throughout plant and animal evolution that are responsible for all sorts of interesting and important adaptations. Seems to be a general feature of eukaryotic evolution to be able to bring inside of cells other organisms and keep them there rather than eating them. Keep them there and use them for some purpose. There are also many cases, we will talk about some of these, where plant and animal hosts have engaged in a symbiosis with bacteria and or archaea. They don't actually bring them inside their cells, but maybe they bring them inside the organism, like into the gut of ruminants. There are lots of various microbes in the gut of ruminants that are used to help with cellulose digestion. So this is sort of a rule of eukaryotic evolution. Lots of symbioses. Multicellular eukaryotes in particular engage in lots of symbioses with microbes, including microbial eukaryotes and microbial bacteria and archaea. It's probably related to the fact that it's just very easy for them to engage in this symbiosis, and it means they don't have to invent a new process, they just have to find an organism that carries out that process. So are there questions about these primary and secondary and tertiary symbioses before we talk about another form of acquisition and merging with lateral gene transfer? So one thing I didn't mention, which you should also think about, is the endosymbiotic events that I talked about are an example where it's useful to think about multiple parts of a genome evolving together within one lineage. It's similar in concept, although for a very different purpose from the discussion we had of duplicated genes, the elongation factors, and it's going to be very similar in concept, although again different in purpose, to discussion that we're going to have now of lateral gene transfer. Okay. So I just want to spend the last few minutes here talking a little bit about what goes on in particular in bacteria and archaea. So bacteria and archaea appear to have a difficult time with bringing other microbes inside of their cells. That's partly their cells are small. Eukaryotic cells can be enormous. That's partly because many of them are surrounded by a hard cell wall. So they don't have an amoeboid form, and they don't eat by phagocytosis. They eat by other means, and so there's just not generally a lot of bringing other organisms inside their cell, either to eat them or to eventually engage in some type of symbiosis. And that means that they're constrained in terms of their ability to acquire functions via this route that many eukaryotes take, which is symbioses. And it turns out that bacteria and archaea have open to them an alternative means to acquire functions from other species, and that's something known as lateral gene transfer, where they take DNA from another organism and incorporate that DNA into their genome in some way. And I'm first going to give you an example or two of this, and then try and talk about what this means in terms of evolutionary history. So the classic example of this, and one that everybody here should be at least a little bit concerned about, relates to antibiotic resistance. So I assume you're familiar with the notion that if you treat bacterial infections or bacteria in the lab with an antibiotic, Those antibiotics are designed to kill certain groups of bacteria. Over evolutionary time, bacteria can evolve means to become resistant to those antibiotics. That's through some type of genetic change in their genome that leads to them to either pump out the antibiotic out of their cell or change their cell features such that the antibiotic doesn't attack them anymore, or they can have degradation pathways that chew up the antibiotic. There are many different means by which a microorganism can become resistant to a particular antibiotic. There's a lot of concern in normal treatment of infections in humans and animals and even plants that by overusing antibiotics, we are going to lead to the increased spread of antibiotic-resistant versions of many different species, and this is certainly occurring. So MRSA, for example, methicillin-resistant Staph aureus, is very nasty. You have an infection with this, it's resistant to many of the antibiotics we throw at it. There are versions of tuberculosis and Klebsiella and many other bacteria that are resistant to various antibiotics that we throw at them. There's a second complication, which is that antibiotics are also used throughout agriculture, and now people are putting antibiotics on walls and on kitchen counters and on your phones and in your cars. And wherever we put these antimicrobial compounds, it will lead to selection of resistant strains of bacteria in those environments that now can resist those particular antimicrobial compounds. It's not that big a deal if those organisms don't ever infect organisms that we care about. But there are two reasons to worry. One is that if we treat animals and breed antibiotic resistance in animals, many of the bacteria that infect animals also infect us. So those antibiotic resistant strains in pigs can move into humans and make us sick. But possibly a bigger concern or an alternative concern is that bacteria can steal the genes that make one species resistant to antibiotic and spread the antibiotic resistance to species that have never been resistant to that antibiotic before. So if you treat chickens and pigs and other farm animals with antibiotics and they have a particular species of bacteria that doesn't even infect humans, but it becomes resistant to antibiotics, if that bacteria encounters a bacterial species that does infect humans, the one that does infect humans can steal the genes from the other one and thereby lead to an antibiotic-resistant human infection. And this stealing genes from another species is generally known as lateral gene transfer. There's another case that I think was discussed in the lab that involves the bacterium E. coli. If you take many different strains of the bacterium E. coli and you compare them to each other, you see a lot of genetic similarity. That's basically what defines E. coli as a bacterial species. If we do the same thing with primates, we compare the genomes of humans, chimps, and gorillas, we see a lot of similarities. That sort of is what defines genomically primates. However, if you look in parts of each of these E. coli strains, you can have, this is a Venn diagram showing the overlap in gene content between three different E. colis. So these are the same species of bacteria with thousands of genes that they share in common. That's in the center of this Venn diagram. But strangely, each strain of E. coli can have up to a quarter of its genome be completely unique compared to other strains of E. coli. We don't see things like that in humans. If we compare all the people in this room, we're going to basically have the same genes as each other. We will have lots of polymorphisms within those genes. We will occasionally, someone will have one gene that other people don't have, but the percentage of similarity between two humans is something like 99.5% of our genome. The percent similarity of two E. coli is more like 50%. The reason that different strains of E. coli have very unique gene content is because of lateral gene transfer. They are able to take up DNA from their environment and make one strain of E. coli have very different phenotypic properties than another strain of E. coli. It's why some E. colis make you very sick. E. coli 0157H7 leads to very unpleasant symptoms and can kill people. And E. coli K12 lives inside the gut of probably everybody here and actually is beneficial to us. And the reason for these differences is strains acquiring DNA from outside sources. So I'm just going to, don't worry about the text here, I'm just going to take you through in the last few minutes just a model to show you how to think about the occurrence of lateral gene transfer in terms of inheritance in bacteria. So bacteria and archaea, most of the time, most species divide by a process known as binary fission. You start with one cell, makes a copy of its genome, and then it splits right down the middle where one daughter cell gets one copy of the genome, the other daughter cell gets another copy of the genome. This is, sped up, 50 or 100 times. This is basically many versions of bacteria just do this. They grow and then split and then grow and then split and so on. And binary fission, if all else is normal, if you look at a cell, it produces daughter cells, those produce daughter cells, and you build, basically it's like an evolutionary tree, but it's a cell lineage tree of inheritance of the genome across these different cell division events. If you extend that over thousands to millions of years, you can in essence get the evolution of species, if not all else is equal. And a really important thing to remember, and again, this goes back to the mitochondrial and chloroplast story and the gene duplication story, is that inside each cell, there are thousands of genes. The genome needs to make copies of these thousands of genes, but if all else is normal, the daughter cells will inherit the cells from the parent in a process of vertical inheritance. Occasionally, you will get a mutation so that you have a new type of cell here and a new type of cell there. This is what leads to polymorphisms in populations that reproduce by asexual reproduction. Occasionally, one of those lineages will die out and another lineage will reproduce more than the others. This is this asexual reproduction that occurs in the basic mode of reproduction in bacteria and archaea and during the asexual phase of the life cycle of eukaryotes. Sorry, just the text here is not really relevant. I'm just trying to explain this. So in eukaryotes, one thing that happens that's different than bacteria and archaea is different lineages can combine together by sexual reproduction to lead to hybrids between one lineage and another. That's the general process of sexual reproduction. We'll talk in a lot more detail about the fine-scale details of this with meiosis and so on later in the course. Requires some type of meiosis and fertilization. This recombination does happen in bacteria and archaea, but it's very different. It's not two lineages merging together. It's one lineage, the DNA from one lineage, moving into the genome of another lineage. This is the process of lateral gene transfer. So it's DNA, not the whole genome here moving into another cell, not them merging to create a 50-50 hybrid, but usually small parts of the genome of one cell moving into another lineage. And that creates a chimeric bacterial or archaeal group. And if you build evolutionary trees after this lateral gene transfer event, we see something very similar to when we look at evolutionary trees of chloroplast and mitochondria and nuclear genes in eukaryotes. Sorry, just going to skip ahead. So what you see is that the core of a species, you can build evolutionary trees of many of the genes in a species, and they all follow the same pattern. This is a figure from the book, and you should read the little discussion on lateral gene transfer in the book. But occasionally, one gene will move from one lineage to another. If you build an evolutionary tree of just that gene, it will show a pattern different from the evolutionary trees of the rest of the genes. So if DNA from the C branch moved into the D lineage, what you will see is that C and D will group together more closely to each other than B and C do, which is for the rest of the genome. And this discordance, this fact that you see some genes with different evolutionary histories than other genes, in eukaryotes is primarily the result of these endosymbiotic events. And in bacteria and archaea, it's primarily the result of these lateral gene transfer events. And I will stop there.